Okay, I'm sure everybody knows all of this, but just a brief overview of what happened. April 20th of last year, the worst oil spill in the nation's history. There have been several that exceeded it, but certainly the worst in the nation's history. Almost 4 times the size of Exxon Valdez in terms of the sheer amount of oil that was released. It went on for an unprecedentedly long time, approximately 100 days before they had it, officially stopped, and then it was formally sealed in July 15th. Where's the little light button? There we go. It wasn't actually capped until sometime early in September, I believe. But in the intervening time, almost 100 days, a very significant amount of oil was released into the water, approximately 4.9 million barrels. The exact split between how much of that was actually oil and how much of it was gas, methane, and other hydrocarbon gases is uncertain. It appears to be about a 50-50 split. But in any event, that's a very significant amount of oil that was released into the water, almost... I should say 200 million gallons. 200 million gallons of oil. One of the questions that I always get is, how bad is this? How bad is this? How bad was it? And there's been a lot of data. The oil was degraded. The oil was evaporated. The oil was dispersed. It's not that bad. And very broadly, you can say that we got off very, very lucky. Things could have been much, much worse than they were, but there's simply no way that releasing 200 million gallons of oil into the water near some very sensitive breeding grounds is anything but bad. It's only a question of how bad does it end up being. And I do think we ended up being very, very lucky. It could have been much, much worse. But here's a picture of what it looked like early in July. And this is a map of some of the landings that we've reported. You can see the majority of the oil made it ashore in the Mississippi Delta in Louisiana, but there was still a significant number of sightings and measured incidences along the Mississippi, Alabama, and Florida coasts. This includes things like verified oil sheens in the water, slicks in the water, and tar balls washing up on the beaches. And one of the things that I want to kind of hopefully get across to you guys, the idea that oil isn't oil. Oil is a very broad term, and it means a lot of different things in different situations. For example, the oil that's here, this sweet light Louisiana crude, means that it has differing sulfur contents, differing densities, different fractions. Oil is a mixture of hundreds of different components, and some of them are of concern from a toxicology standpoint, and some aren't as much. The ones that are of probably the most concern for us, I'm an aquatic toxicologist, what we call the PAHs, the polycyclic or polynuclear aromatic hydrocarbons. These are the ones that are known to be associated with carcinogenesis in vertebrates. It can cause acute toxicity, it can cause chronic toxicity, it can have effects multi-generationally. And so they're the ones that we're really most concerned about. But they're not the only ones. There's a very light fraction of oil that's called BTEX. It stands for benzene, toluenes, ethylenes, and xylenes. One of the things you'll notice as I go through this, and I'll have a slide on it later on, scientists love acronyms. Anything you make an acronym out of, we always like to do. But BTEX is one of them. And some of the other components are the ones that are going to hang around longer. But the PAHs are the ones that are kind of in the sweet spot. They're not immediately degraded or immediately volatilized, and they're probably of the highest toxicity. So they're the ones that people tend to focus on. But one of the things I hope to get across to you is that the concentration of pHs in the water or in the sediment changes as a function of time. As they move away from the oil spill, the relative concentrations of these different components is changing over time, and that's directly affecting the toxicity that organisms exposed to it will be seeing. When you add in the effect of Corexit, as I'm going to talk about in a few minutes, you can see that it becomes even more complicated. So one of the people always ask me, why don't we know more? And I'll address this a little bit later. One of the answers is, this is a really, really difficult, complex question. It really is. There is no simple answer. So, some terminology. Like I said, acronyms are everything in science, particularly in toxicology. But these are some ones that you might see in the paper or when you hear scientists talking about it. Whoops. What did I just hit? There we go. Okay, WAF. WAF stands for the water accommodated fraction. This means when you take oil and you put it in water, like happened in the Macondo well, a certain percentage of it partitions or goes into water very naturally, very normally. Now, you'll say oil and water don't mix. It's true, and this is a relatively low percentage of what goes in. Most of it will end up floating to the top as a slick, like we all saw. The CWAF is another one. That's a chemically enhanced water accommodated fraction. This is also known as dispersed oil. This is what happens to oil when you add dispersant to it, like the Corexit that we saw very widely used in the Gulf of Mexico last year. And this works exactly like soap works. when you're washing your hands. If you're cooking dinner or something, you get olive oil on your hands and you use soap to wash it off. It acts in exactly the same way. It forms what we call little micelles, little bubbles of oil, and a dispersant wrapped around it. And the oil is very, it doesn't mix with water very well, but the correct dispersant does. And so you get these little bubbles of oil surrounded by something that mixes with water very well. And you end up with these little micelles or little bubbles floating through the water column. The slit gets broken up. It's not visible. It gets dropped down into the water at depth. And they do this for a couple of reasons. Now, the cynical view is they do it so that you don't see the oil anymore. That's not strictly, that may play some role in it. But the biggest part is that it increases the surface area to volume ratio. A sphere always has more surface area available than a slick does. And so there are microbes that are present in the water that will eat this oil very, very quickly and turn it into more bacteria, use it as a food source essentially. And by dispersing it, by forming these little bubbles, they make it more bioavailable to these bacteria and theoretically enhance the microbial degradation. The problem from a toxicologist standpoint is that they're also increasing the exposure route to fish and shrimp living in the water. In other words, if you've got oil at the slick at the surface and you've got fish swimming around below it, unless they actually go up to the air-water interface, you've got a relatively minimal exposure. You've got some, the WAF, the water content fraction that's normally partitioning into the water as a result of this, and they're exposed to that. But the majority of it's going to be up at the surface and theoretically non-bioavailable. Once you disperse it, though, it's now floating through the water column. Fish are swimming through here. They're encountering these droplets. Perhaps they're eating these droplets. They're certainly getting it on their skin and across their gills, and they're exposed at a much higher rate. And there's very, very good data that dispersed oil is accumulated in fish at much higher rates than the non-dispersed oil is. So it's a trade-off. You're enhancing theoretically the microbial degradation of the oil, but you're also enhancing the exposure of other organisms to the oil. And so it's a balancing act as to which one is happening faster and which one is more important. Another term is weathered oil. You've probably heard this a lot in the media. As the oil is coming out of the well, for one thing, it's coming out at something like 1,200 degrees, and it's hitting water that is significantly colder than that, somewhere a little above 4 degrees Celsius. And so when you've got this really hot oil blasting out under a lot of pressure, high speed, at high temperature, you get a certain percentage of it was physically dispersed. It was blown apart into little bubbles and not seen anymore. You also tend to get preferentially some of these lighter fractions, lighter molecular weight fractions, like the BTEX, benzene, toluenes, ethanol, xylenes. These are fractions that have low numbers of carbons. We talk about oil as a hydrocarbon. That means it's composed mostly of hydrogen and carbon atoms together. So benzene, toluene, ethanol, xylene have 2, 3, 4 carbon atoms arranged in different structures, and that means they're very light. Composed of something like benzoate pyrene, which has 5 ring structure times 6, 24 carbons. So it's much denser, much heavier, hangs around a lot longer. But the BTEX is stuff that's light enough that when it's exposed to sunlight at the surface, it tends to volatilize away. If you guys were here last summer, you might remember occasionally we get this smell coming in, the strong smell coming in from the ocean. That was the BTEX. That was mostly benzenes, but also toluenes, ethanols, and xylenes that we were breathing as it was volatilizing off of the oil slick, the winds were blowing it into shore, and we were breathing it. It wasn't so much the burning effect that some people thought it was. It was just the strict volatilization of the BTEX getting removed from the slick just outside the site. So that's some of the basic terminology that scientists use when they're talking about this. And now I'm going to go on and talk about some, not so much our data, but well, first is another concept I want to get across to you. This one was always hard from a toxicology standpoint to get across to people. People ask me, is something toxic, is it not toxic? And there's never a yes-no answer to this. We have a saying, kind of a tongue-in-cheek saying, but nonetheless it's true. Paracelsus's law, the dose makes the poison. Anything in high enough concentration, a high enough dose, will be toxic. You might remember 4 or 5 years ago, the case of the woman died from water toxicity from the hold your wee for a wee thing they had at some radio station out in California. Well, she drank a lot of water, and they didn't let her urinate. The water in her body diluted the ions in her blood, and she ended up dying essentially of water toxicity. Conversely, the most toxic substance on the face of the planet, various snake or spider venoms, if they're present in low enough concentrations, low enough dose, won't have an effect. So the idea of toxicity in marine organisms that they experienced in this event was directly related to one question. How much oil were they exposed to, and how long were they exposed to it for? In other words, if you want to look at lethality as an endpoint, then you say, they were exposed to a high concentration for a short period of time or a lower concentration for a longer period of time. You can almost look at what we call an integrated dose. Now, this is complicated dramatically by the fact that time directly affects the proportion of constituents that are present in this oil. So as time goes on, the lighter elements are volatilizing out, the concentration of other elements is changing, all of which have different toxic components and toxic properties. And so, again, it's a very, very complex situation. But the key concept to get across here is that dose, mass and time that an organism was exposed to is going to in large part, determine the toxicity that it observed or that it was exposed to. And this is one of the key cellular molecular pathways that gets involved. And I don't want to spend a lot of time talking about biochemistry here, but this is kind of the canonical or the usual example of how an organism responds to hydrocarbon exposure. This is called generally the mixed function oxidase pathway, or in this case the cytochrome P451 pathway. Don't worry about the names. But I just want to make the point that we have all of this in our bodies. It's active constantly. Fish are very, very good at this. Shrimp and crabs and other invertebrates, a little bit less so. They have something analogous to it. But basically what happens is this is benzoate pyrene. This is a PAH. This is something that's known to be carcinogenic, known to be toxic, will kill fish very, very quickly. Just as an example, it enters a cell, human, fish, any vertebrate or whatever. It binds to a protein that's called the aryl hydrocarbon receptor. Don't worry about the names. But the point of this receptor protein here enters into the nucleus, binds to another protein called the aryl hydrocarbon receptor nuclear transporter. They bind to a section of DNA, and they, what we call induce, they cause this gene to be expressed at very, very high levels very, very quickly. This gene is called cytochrome P451A. It moves out into the nucleus and will grab onto these PAHs before they can bind to AHR theoretically and metabolize. They'll cause the structure of the pH to be altered in such a way that the animal can then excrete it very, very quickly. Now, the upshot of all of this is that in animals that have a high-functioning MFO or cytochrome P451 system, it's almost impossible to get high tissue burdens of PAHs. This is so effective that it grabs onto these and kicks them out very, very quickly. Now, that means that getting high tissue burdens that might be of concern when you're eating a fish would cause very high levels of PAHs to achieve that, and it might probably you'd end up killing the fish before you could do that. Now, obviously, every animal is different, every pH is different, and that's just kind of a general rule of thumb. But I wanted to at least introduce you to this pathway that is very, very well characterized, and I think is really kind of fascinating in how an animal is able to very quickly and very efficiently combat the exposure to some very toxic components. And this is something that's evolved over the course of several billion years. You can find analogs to this in animals that are evolutionarily unchanged for 500 million to a billion years. It's present in most animals that we see. Okay, so the other thing that people were always concerned about in this was the dispersion that was used. Correct, they used two different ones. 9500 was mostly what was used. They also used a slightly different formulation, 9527. Effectively, they're the same thing. Tremendous application rates. They've never been used at anything remotely approaching this level before. An estimated 1.5 to 2 million gallons were applied. About 2/3 to 3/4 of that was applied directly at the surface. The remainder was applied at depth, and they were squirting it out of the tanks of the ROVs onto the lake that was emerging directly.